As we have seen with several examples so far, evolution at the molecular level works by combining existing modules, such as protein domains, into ever more complex arrangements so that more sophisticated molecules can sustain changes in morphology or function of the organism. Obviously, this requires the reshuffling of genetic modules. In this regard, the peculiar structure of eukaryotic genes has been crucial. As we have seen in the previous video, very early during the appearance of eukaryotes, genes became split, with exons interrupted by introns, which probably originated from group 2 self-splicing introns from bacteria. Once introns were present, they expanded rapidly in the genomes of all animals with bilateral symmetry. In fact, when scientists compared the genomes of deuterostomes, animals which included tunicate Siona intestinalis, Branchiostoma, the Amphioxus, a lancelet which has an autochord, and other true vertebrates like humans, they found something very interesting. Amphioxus, very ancestral chordate, and humans, who are at the very tip of the vertebrate tree, share 85% of their introns in terms of location of those introns, their position in the genome. So it seems that introns, at least during vertebrate evolution, have been mainly increasing in size. So having genes with exons and introns is a feature that really can explain how genomes have encoded an increasingly more complex proteome. It was something really crucial for genome evolution because it made possible all this reshuffling of modules. But why and how? Well, one of the answers to this might very well come from our old friends or foes, the viruses. Introns, like all that intergenic DNA that links genes in a genome, are very rich in mobile elements. And mobile here means that each element, a short fragment of DNA, can cut itself or sometimes copy itself and then move to a new location in the genome and insert itself there. Mobile elements are roughly classified into 3 categories: DNA transposons, some types of which are already present in bacteria, LTR retrotransposons, and non-LTR retrotransposons. And LTR stands for long terminal repeat, a characteristic sequence located at both ends of a retroviral genome. LTR retrotransposons, as the name implies, come from ancient retroviruses because retroviruses have been infecting animals for millions of years, leaving behind in the genomes of the infected hosts bits of their own genomes, which are known as endogenous retroviral elements. Non-LTR retrotransposons, on the contrary, have nothing to do with retroviruses, although they have a long history dating back 500–600 million years. And as I say, they do not seem to come from viruses. Some of them are similar to ancient RNAs and code for a retrotranscriptase similar to some group 2 introns. So we find all these types of mobile elements in the genomes of all eukaryotes. Some species of yeast, for instance, have the TY and TF LTR elements. Dictyostelium discoideum has the DERS1-like family of elements, which show some features of LTR and some features of non-LTR retrotransposons. Drosophila has non-LTR retrotransposons, and mammals have endogenous retroviral elements, ERV elements, as well as non-LTR elements like Alus, a family of short dispersed retrotransposons which are derived from a very ancient RNA called 7SL RNA. In mammals, these families have expanded very efficiently because in flies and fish there are many classes of transposable elements, but each class is found only in a few copies in the genome. They account for not more than 5% of the sequence of the entire genome in these species. Whereas in mammals, we have fewer classes, but thousands or even hundreds of thousands of copies of these elements. In fact, they have grown so much that in humans they comprise almost half the sequence of our genome. This, as you probably know, was formerly called junk DNA. Today we know that it is no junk, and that it has actually helped to shape our genome in many different ways. How? Let us see.